Thanks so much, Dan. It's a really great pleasure to be here and to tell you some of the things that we are doing. And in fact, this project is a project that's a result of a collaboration with Dan, trying to isolate circulating tumor cells, where Dan has long interest in. So in fact, one of the things that we started out to do is to try to develop a technique to isolate rare cells in general from the blood. Blood is another organ, and there's got a lot of cells that are present at very few copies, few numbers, copy numbers, and we were interested to see if we can develop a method to isolate those cells. And I think that with the ability to isolate very rare cells, then there are sort of more rare cells that comes about that one can learn about, and so on and so forth. But the circulating tumor cell is the first one that we started working on together with Dan, because there's been a lot of interest in this area, and also there's been quite a bit of groundwork that had been done in this area, and I'll talk about that in just a little bit. The basic idea, this was actually first started by a group and also company, it's called Viridex, which I'll talk to you about, where they use a machine and they saw that if you can isolate circulating tumor cells and count the number of them, is actually got some prognostic value. And this is how everything got started, because those cells are rare, and many people thought that, well, if they can isolate a few cells, are there really just a few cells, or maybe perhaps there are more, and it's only that the techniques were not as well, is not as sensitive, and therefore they only can isolate a few cells. So we thought about the same thing together with many other people, and so this is how our technique got developed. And so just to give you a really quick snapshot of trying to look at this technical challenge of isolating rare cells, what does rare cells mean, and how many cells are there in blood. So you're a pathology audience, so I think you probably know this better than me. But the bottom line is that if you look at Viridex system, the threshold to say whether a patient has a good prognosis or not so good prognosis for metastatic breast cancer, and I was told to use my cursor rather than a pointer, so let me see. Okay. A metastatic breast cancer is only 5 circulating tumor cells in 7.5 ml of blood. And in 7.5 ml of blood, you can look at the number up there. You're going to get tens of millions of white blood cells and tens of billions of red blood cells. So out of those tens of millions of white blood cells and billions of red blood cells, there's only a few circulating tumor cells based on Viridex technique. And so the challenge is that, well, out of this such large background of blood cells, can one isolate those rare cells or not? So this is sort of is the technical requirement. And so the first thing I want to tell you is that, well, there are certainly a lot of different ways that you can think about doing this. And so the way that Viridex developed, and a lot of things are benchmarked against Viridex, because they are the only FDA-approved system out there that that's available. And so the way they do it is that they say, hey, okay, those cancer cells would have some cell surface marker that are unique to them. I'll come back to this. And so if they would have magnetic beads that are small means 100, 200 nanometer in diameter, that has the antibodies against a cell surface receptor, then they can mix it together, and then they can use immunomagnetic isolation to purify it. And then afterwards, the purification, then they would image them. They would basically image a large area, collect those images, and then do additional biomarker imagings and so on and so forth, and then you can determine how many circulating tumor cells there are. So this is the approach. And actually, in fact, Dan has a machine that we used in the collaboration. I'll show you some results. So this is the approach. It takes about half a day to do the auto prep, and then you also have to take time to screen through a large area for those cells and then look through them and so on. So it's not a particularly very easy thing to do, but you can do that, and they're the first one to do this. The cell surface marker they use is EpCAM. So in fact, that is an epithelial cell surface marker, not a cancer cell surface marker. But the idea is that, well, most of the cancer cells would be epithelial in origin, and so maybe that will be fine. So this is a marker that they're using, And when we do our work to compare with their system, we use the same marker. But obviously, I think one of the things that will be very interesting is to look at other markers that may be more unique to the particular cancer that you're looking at. So this is how the field was maybe 5-7 years ago. Since then, because it's a very clear technological challenge, there has been quite a lot of ideas out there to try to develop different techniques to isolate those rare cells. I won't go through them in great detail, and because we can talk about half a day about pros and cons about different techniques. But I usually try to categorize them into different strategies. So the first one is that, well, you can use magnetic particles or some kind of particles that binds to the cell, and then you can pull out those cells because you have the particle that bound to them, and the particle acts as a handle. So this is what Viridex do. There are a lot of different versions. There are groups that try to improve the magnetic field to see if they can make it more efficient. There are groups that are trying to do, rather than positive selection, meaning that cells that express those receptors or proteins, rather than doing positive selection, would do negative selection, meaning depleting cells that have a certain marker like CD45. There are another group of techniques that is use devices called microfluidic systems. So what they would do is that they would put antibodies on the surface of the microfluidic channels. And the idea is that, well, when the cancer cell flows through the channel, they interact with the surface. And if they don't have the cell surface receptor, they won't be bound by the surface. And if they do, then they will be pulled off from the solution and then bound to the surface. Then you can go in later on and try to image them and so on. So there's a group of people that do that. And they have pretty good results also. Another group is to use imaging to try to image through a large number of cells. And then finally, the last one is to try to, rather than use cell surface markers, to use the fact that circulating tumor cells, what people say is that, well, they are larger than blood cells. And so if you can separate something by size, then you can maybe pull out those cells based on size rather than based on markers. And that's kind of appealing because, well, then maybe you don't have to worry about whether the cell has that EpCAM marker or not. So these are just to give you some background flavors, and you can really go quite deeply into sort of looking at the pros and cons of different technical stuff. But just to give you a survey, because there's a lot of stuff out there in this field, and it's a very active field, and just to give an overview of this. So the way we started actually initially was to look at size, because it was kind of appealing to us that you can use size to isolate circulating tumor cells. So what I'm going to tell you is some of the things that we do that based on size separations very quickly, and then show you that, in fact, I think that the more recent thoughts that circulating tumor cell is in fact very heterogeneous, especially if you look at patient samples. And so there are actually small circulating tumor cells, and this may not be the best way. So I'm going to give you a sort of a story about how our technique came about, how it incorporates size, and how it evolved over time to come up with a final platform. So initially, we tried to do size, and what we did at that time is that instead of going to a filter, like a polycarbonate filter that you would use in the lab, we would actually use microfluidics. Our lab do quite a bit of microfluidics. So we use microfluidics to engineer filters that might work better for this purpose. And so the filter that we have developed, we call it a one-dimensional filter, actually is nothing more than sort of slits, arrays of slits arranged in a particular geometry. And this works pretty well because if you look at red blood cells, the red blood cells is a disk, and it turns out that when the red blood cells go through, encounter the slit, they would actually all turn on the sides to go through the slit. And so this is a video just to show you. So that is kind of nice that you have a hydrodynamic force that actually turns the red blood cells on the side, and it's very easy to go through the slits. And also, red blood cells are very flexible. They're very good at going through very small spaces anyway. The other thing that turns out to be quite good with this design is that if you have a cell that blocked off the slit, well, the slit is not completely blocked off. The fluid can flow around the cells, and as a result, the kind of hydrodynamic shear stress, the hydrodynamic pressure that you exert on the cell that you are interested in and that is retained by the filter is actually quite mild. So we did an analysis simulation, if you have a hole and the cell completely occludes the hole, it experiences a lot of pressure. But if it's a slit, it's much less. Much less meaning, if you look at this scale, there's a log scale on the right here, it's actually orders of magnitude less. So that's very nice, because one of the things that we've been really interested in is to isolate those cells and then do further things with those cells so that we want to have those cells be viable and so on. So this is what we designed in terms of the slit itself. And then to do filtering for whole blood, that's a lot of cells, and there's a very dense number of cells. So we develop microfluidic systems to do that, to try to handle that. And so we created something we call effusive filtration. So it's a particular microfluidic design that we have that we will flow blood through, and then it will go through a system like this, and then the cells will get trapped. And that turns out to be, we went through a lot of iterations and simulations and device making, and we found this works pretty well. And so we went ahead, and then we went to try to see if we can find some circulating tumor cells from samples that Dan gave us. And so we would filter blood. You would just have blood going through. And in fact, we found that we could, using this method, to isolate circulating tumor cells from patients. And we see some cells that are really large. But also, when we use other techniques and so on, we also see cells that are quite small. So the bottom line, I think, is that at that point in time, we were really worried that we would be missing some of the key circulating tumor cells if we simply rely on size. But anyway, what we did is that we did characterize how good the system is using spiked-in culture cells, so we have a model system. And it turns out that if the cells are fixed, so they act more like beads, you can get pretty good retentions and recoveries. If they're alive and they're more deformable, then it's a little bit worse. But it works pretty well generally. But anyway, we were worried about that we are going to lose a lot of the smaller rare cells that might be really important. So what we ended up doing is to use filtration, but only as part of the system that we use to isolate the cells. We have another part that relies on cell surface markers, so we can use markers to pull things out too. So we can combine those two, and hopefully we can get better performance. So the platform that we developed is something we call eDAR. And so this is, I'm going to go through the concept first, and then I'm going to go through each pieces just to give you some idea for how each pieces of this device works. So the basic idea is that you get a lot of cells in blood, and you have to find a few cells that you're interested in. So if you want to look for single cells, one of the techniques that is commonly used, of course, is flow cytometry. If you try to apply flow cytometry to rare cells like circulating tumor cells, it doesn't work very well because the throughput is not high enough, and then the sensitivity is not high enough. So what we have decided to do is that, well, instead of looking one cell at a time like a flow cytometer, where we know the throughput is not good enough and it's hard to sort when you go through so many cells, how about if we look at it instead of one cell at a time, one aliquot of blood at a time? So we actually take the blood and then we divide it into aliquots. And then we can ask ourselves what is the optimal aliquot and so on and so forth. We can talk about that. But anyway, the basic idea is that we take whole blood, instead of looking at one cell at a time, we will be looking at aliquots of blood at a time. And the aliquot that we use currently is 2 nanoliters. So each aliquot will have about 10,000 cells or so. Okay. And so this is what we do. We would then use a microfluidic here initially to make aliquots. So if you want to look at and screen through aliquots, you have to somehow define an aliquot or make an aliquot. So I'm going to go through that. And then we do very sensitive laser detection, line confocal detection. Anyway, the end result is that we have multiple color lasers and detections, kind of like a flow, where we can see if the aliquot contains a rare cell or not. If it does contain a rare cell, then we would direct it to this channel that will collect this aliquot. Most of the aliquot won't have any rare cells that you will be interested in, so automatically those ones will go to the waste. So it's a very efficient enrichment technique because 99%, 99.99% of the aliquots will be going to waste automatically. And then a few aliquots or a few tens of aliquots or 100 aliquots that you are interested in will be directed for further separation analysis. So it's a very useful enrichment method. It's analogous to flow cytometry a little bit, but it's really quite different. For example, some of the things that you can get in flow cytometry, you cannot do with this at all. So flow cytometry, one of the reasons it's very powerful is that you can look at scattering signals, to look at shape, to look at morphology. You can do a lot of on-the-fly analysis, but this you cannot because there's so many cells. But what you do get is that you can collect the aliquots later on, and then once you have those aliquot and the few cells that you're interested in, then you can actually study them with really high precision and so on and so forth, much better than you could with the flow. And I'll show you some of the things that we're doing currently. The other thing is that if you think about this technique, this really works well when you have rare cells. If the cells are not rare, then every aliquot would have a cell, and then you won't be doing anything with this device at all. So it works really well with rare cells. You have very high throughput. Right now, we can go through 1 ml of blood in 20 minutes, and we can make it faster. So I think the throughput is actually really good. So anyway, the aliquot still contains 10,000 cells in addition to your rare cell. So what we do then is that we send it through a filtration chamber, like I told you before, to get rid of most of the red blood cells and some of the white blood cells. But the filter size slit we use, or holes that we use, are really small, 5 micron, 8 micron, so they're not really small, but they're small enough such that we're not going to be worried about losing small circulating tumor cells. So this is the overall strategy, and I just want to show you, some of the pieces that we have, and then more importantly, tell you how it performs and what it can do. So the first thing that we did when we make aliquots is actually we do something called droplet microfluidics. So what we actually do is that we take blood, and then in microfluidics, you can have ways to put the blood, aqueous is sort of aqueous-based solution, together with an immiscible phase, so that's oil. And because of fluid instabilities, it would actually make droplets, and you can make droplets really fast. We can make droplets up to 10,000 droplets per second. So you can really make them really fast. So that's not the issue. And so you can see that there are whole droplets of blood here, and then these are the oils, and you can flow them down. So this is the initial things that we do, and it kind of was very appealing conceptually, but it turns out that it didn't work quite well for us. And the reason is because this is very nice when it worked really well. You have a stream of droplets, but it's very sensitive to conditions. So it's kind of like a traffic jam. If you have anything that happens downstream and you have an accident, then your whole trip goes to hell, kind of. So that didn't work very well. The other thing that I thought this would work really nicely is because, conceptually we thought, hey, it would be really nice if those rare cells are protected in this droplet, and then it's not going to experience all those shears and stuff like that outside the fluid. And that would be really great if we can do that, because then we can isolate those cells. They're not so stressed, and we can do a lot of stuff with it. And that turns out not to be entirely true either. So we did a pretty systematic study, and we found out that actually in droplets, you can have very interesting recirculation flows. So there's actually flow inside the droplet, so that the cells can actually experience quite a lot of shear in many cases. And in fact, I think that if you make those droplets fast, and if the conditions are, with fast floats and so on and so forth, we can even see that sometimes the cells can pancake against the interface between the aqueous solution and the oil. And we have studied this quite a bit, so I'm not going to dwell on it, using different ways to make droplets. So we sort of scratched our head a little bit, and then we decided that, well, actually, we don't really need to have a physical droplet to define the aliquot. We can actually use our laser beams and the way that we sort downstream in our illumination areas and so on and so forth to define a virtual aliquot, because it doesn't really matter if they're physical or not. So we decided then to go with this notion of a virtual aliquot where the blood actually is a continuous flow, but we define the aliquot using our detections, our illuminations, and so on and so forth. And that turns out to be really nice, because in that case, we don't have oils and droplets, and we just have blood flowing through a tube, and that was extremely robust. The second piece is that now we have this cell in 10,000 white blood cells, are we able to see that there's one cell in 10,000 background cells? And it turns out that we can if we do very sensitive detection. One thing we do quite a bit in our lab is to set up optical systems so we can do single molecules of dye detections and things of that sort. So we optimize our optical detection and so on and so forth. And we saw that at the moment at least, we saw that the best of aliquot number to use is around 2 nanoliters because we can detect one cell in that background. And here just show you, the cell that we can detect in flow. And here show you that if you have a really dense blood, and then you cannot see the cell at all because it's buried in there, it's extremely dense. And if you do sensitive detection and so on and so forth, you can in fact see them quite well. So the point we are at this point is that we can actually use virtual aliquots, and we saw that we can detect one rare cell in the background of whole blood, even though you have a very dense blood. So we can do both pieces. The third piece that we had to do is that, well, once we see the aliquot, we mostly let them go, but the occasional time we see an aliquot that contains the rare cell that we wanted, then we want to sort it. And in fact, we call it aliquot ranking because we can do quite sophisticated logical operations like, hey, maybe we want to see if this cell contains this marker and not this marker. So we can in fact think about all of those aliquots having multiple outlets and then rank those aliquots and direct them in different ways. So you can do quite complex things, I think, down the road. But at the moment, our ranking is 0 and 1. But then 1 as being there's a rare cell, 0 as being there's no rare cells. But I think that later on you can do quite a bit of things with it. And so the sorting part, what we have done is that we develop a sorting strategy that we can sort with about millisecond resolution right now. And this just shows you we have blood coming down, and then you can sort them. I won't go through the details, and we're sort of getting things, improving this part also at the moment. But even at this point, a couple years ago, we can sort really nicely. So what we did was that if we go back to here, we park our detection here so we can decide which aliquot to sort. And then we can park another detection spot here to see if it's sorted the aliquot that we wanted to sort. So we have confirming detection here and then the triggering detection here. So every aliquot that we wanted to sort, we can see on the fly whether they have indeed been sorted. And when we analyze the first channel, when we see something, we say, hey, we want to sort this aliquot. It's got the cell we want. We see that it always gets sorted. And so that works pretty well. That depends on the flow rate. And so at moderate flow rates, this is essentially 100%. But for most of the clinical samples, we want to go and process it faster. So when we do that, it's around, at the moment, it's around 93, 95% or so. But that's pretty good. And this is just shows you in real time what it looks like. We can just direct the flow in either channels very quickly. This is taken with a fast camera, so these are on the orders of milliseconds. So what we have, once we have all those pieces, the platform that we have at least a year ago is this. Now we have a new version now. We have we have the next generation now. But anyway, with this system, what we have is that we have whole blood that is flowing through. We have virtual aliquots. We can do 3-color detection, so we can look at 3 markers. And then we can confirm whether we have sorted that aliquot on the fly, so we know if there's issues. And then we will send it to a cell capture chamber. And this cell capture chamber is only about millimeters square. So it fits pretty nicely with a field of view of a 10x objective. And that is important because many techniques require you to sort of search for the cancer cells over a large area. Here, everything is a very small area that matches pretty well with a lower magnification objective. It's high magnification, you can just do a couple scans. And the other thing that I want to note is that this cell capture chamber, in fact, is open from the top. So here, there's a hole here, there's a reservoir here, the flow goes in, but this is open at the top, so it's really easy to add stuff to this chamber because the chamber with those filters retain the cell. So if we do 3 markers on the fly, we can in fact do more markers on this chamber because we can just pipette in additional markers of different color. And in fact, if you want to do multicolor imaging, sometimes you can do more than 5 easily. Well, in that case, we found that what we can do is that we can look at a few, and then when we're done, since everything in a small area is very easy to keep track where the cells are, we can just bleach them, add a new set of markers of the same color, and then look at them again. So you can do things like that. And also because it's open from the top, I'll show you later on that we can in fact go ahead and remove those cells to do additional things with it. I won't go through this too much. This is just shows you some data about the sorting efficiency as a function of flow rates and so on and so forth. The other thing with a rare cell is that you really don't want to manipulate your sample too much, because if you're going to have to send it through a per-call gradient or any of those, any manipulations that you do is potential for losing the cells. So the procedure that we have is actually quite simple. We take the whole blood, we add the antibodies to label them. Those are fluorescent. And then we incubate it a little bit, and then we just dilute it, do just one spin, remove the supernatant. When we remove the supernatant, we sort of leave half, equal volumes of this in the supernatant behind so that we don't accidentally remove any rare cells, and then we run it through the chip. So it's fairly minimal sample manipulations. So with all of those, I think the end result is that, okay, well, that's fine, but how does it work, and is it going to be useful or not with the sensitivity that it has, and so on and so forth. So the first thing we did, obviously, is take a system that we do know, and then try to see how well this technique performs. So what we did was to take cultured cancer cells. So these are cultured breast cancer cells. It's either SKBR3 or MCF7, those type of things. And then we would spike in those cells into whole blood. And we have a technique, we developed a technique that allows us to spike in precisely a certain number, 57, 62. We have a very precise way to spike in, so we know exactly what we put in. When it is in, then we go through the whole procedure. We would label them and all of those things and run it through the chip. And then when we did that, what we found is that at the flow rate that we use, which is 50 microliters per minute at the moment, we have about 93, a little bit better than 93% recovery. So if you have one cell in a background of billions of cells in this ml of blood, then 93% of the time we can isolate that cell back out in this very small area, so for you to do additional studies. The other thing is that because we can do multicolor imagings and additional biomarkers and so on and so forth, of all the runs that we have done, we have never seen any false positives. So we use healthy donor blood, and we have, if we don't spike in cells, then we don't really see any of those. The other thing that we did together with Dan and Karen was that, well, that's our culture cells, and most of the things you're interested in are patients. Patients are obviously, cells are very different, and so how does it perform in a clinical, with actual patient samples? And so what we did was that we got samples from the patients, Dan gave us, and I think there are 3 tubes. The first tube I think was discarded because they were about contamination from the puncture, and then anyway, the remaining tube, one goes to Karen. Karen runs it, and then the other tube comes to us, and then we will run it. And so when we do that, there's 2 things. One is that we use markers. They're very similar to Viridex. So Viridex use EpCAM. We do that too. The patient that we have is HER2 positive, so we decided that we're going to look at HER2. Might as well look at HER2 as well. They use CD45, so we use that also. And then they use cytokeratin, Viridex, and we use that as well. They use a nuclear stain, and we use a nuclear stain. And so we can do a comparison. And when we did a comparison, we found that in many of the patients, metastatic patients, Viridex don't see any of the circulating tumor cells, and that's, I think, is not surprising, because I think that's sort of been reported. But we see circulating tumor cells in all cases, and when we do see them, we tend to see about 10 times more. So we're pretty excited about that. The other thing is that we can do many markers and so on and so forth. So we decided that since we can pull out more circulating tumor cells, then can we actually look at some subsets of those circulating tumor cells? In particular for breast cancer, it's been studied that if the cell expresses CD44 plus and CD24 negative or low, then they have stem cell characteristics. So in fact, we have been able to see that from patient samples as well. So we're pretty happy about the performance. And just as a side note, we actually collected a lot of data over 3 years together with Dan and Chenghua, some DNA markers to see if we can correlate. So we were excited to be able to, in the future, try to correlate some of those with the amount of circulating tumor cells. The other thing is that Viridex and many of the state of the field at the moment is to count the number of cells. And if you have more circulating tumor cells, you have a poor prognosis. If you have very few circulating tumor cells, you have a better prognosis. But the number, I think, if you talk to, if you think about it, is probably not as interesting as trying to understand the molecular biology of those cells. Are all the circulating tumor cells the same? How does it reflect the primary physiology of the primary tumor? All those kind of stuff. So that's what actually our lab is sort of excited to try to pursue, to be able to do more analysis, molecular profiling of those circulating tumor cells. The other thing that we would like to do is to be able to pick out those circulating tumor cells and then culture them and then create a clonal expansion. And there are a lot of reasons for doing that. If you were able to culture them, then some of the techniques that may not have single cell sensitivity, like proteomic type stuff, then you can actually grow up the cells to do an analysis. You can imagine that if you have actually the patient cells, then you can actually see how they respond to drugs and so on and so forth. So we're very interested to do that. And so we did that with cultured cancer cells. So we spiked in we can spike in cultured cancer cells in blood, go through the whole procedure, and then go in and use a micropipette to say, hey, this is a cell in this field of view, this is a cancer cell, I want to remove it. So in fact, we can remove it, a cell, and then we can grow the cell back. It takes a little bit longer because it's a cell in isolation, and then we can grow the cell back. So this also, I think, will be really nice, but this is so far with culture cells. So if you have one culture cell with 93% of the time, we can not only isolate it, but we can actually grow it. But we try a little bit with patient cells, and that is actually much more tricky because, again, the patient CTCs, I think they are very heterogeneous, and maybe a lot of them are, in fact, maybe apoptotic. So we're still working on that. So as a summary of this part, I just want to sort of recap some of the things that we would like to do and what this sets up, some of the future directions. We would like to do culturing and do clonal expansions. We would like to do functional assays on those isolated cells. I think that would be quite exciting. Apoptotic assays, things of that sort. We are very interested in doing gene expression because that is something that is easy to get single cell sensitivity because you can amplify the DNA. In fact, in the next piece of the talk, I want to just show you some of the technologies that we're developing towards doing that. And also, for different cancers, it's not one disease, it's many diseases and many different pathways gets affected. So we're very interested to do more systems level analysis of pathways and so on and so forth to see if we can develop tools to do that. The fact that you have more sensitivity, I think, is quite nice. There also creates questions. If metastatic breast cancer is 5 for saying whether the patient is going to do well or not based on Viridex assay, we don't actually quite know what that number would be for us because we can see more. But in any case, intuitively, it seems like, well, if you can isolate more, then you have more dynamic range to make certain clinical decisions. At least that's our hope. Because if you think about it, 5, and for, I think, colon cancer or prostate cancer, it was even 3, that's really going down to the noise of stochastics, right? If you see 4, what does that mean? If you see 2, is it because when you do your test that you just, you actually, if the next time you do it, you might have 4, but this time it's 2 because you missed 1. So you're going down to the noise and the stochastics. And the way that Viridex got around it is that they have a lot of patients, hundreds, and then they average them, and then they got an average value. But if you only got one data point, I think that's kind of hard to make a decision. But I don't know. So we still have to find out about that. We're able to see the cells in more patients samples, so we're hopeful that we can hopefully, maybe to go to earlier stage or perhaps expand the number of applicable number of patients. And also, we're trying to start, hopefully, look at, what are the differences between circulating tumor cells? Are they all the same? Are they all different? At the very least, it seems like there are, subtypes, and if you use markers. But there are a lot of things, I think, to look for there. And this is circulating tumor cells, so we're also interested in other rare cells as well. Fetal cells will be very rare. We're very excited. Those supposedly is even more rare than circulating tumor cell, but if you can isolate them, that fetal cells circulating in blood, that will be very exciting. So there's a number of things that that we are thinking about. And part of the thing with this talk and talking to many of you is that it's really very fruitful for somebody like me to develop technologies and get feedback and say, hey, maybe you can think about this and that. Because in the end, we want to be able to translate this to make it useful. And speaking of which, we have trying to sort of prep ourselves so that we can disseminate the technology more broadly. And so we've been working on actually chips that are very clinically compatible, biocompatible. So we designed a new substrate, developed a new substrate we call Puma, that is actually very biocompatible. We can make very nice features out of. So we're transitioning to chips that is very fast to make, is very easy to make. So when I say, hey, we're going to the, from what we just published, we actually have a 2 generation down in terms of the chip design and so on. Most of those is actually try to simplify it. Rather than taking many hours to make a chip, we want to be able to mass produce chips, and so that we can actually use those chips, toss it with very minimal labor and cost. So I won't go through this part too much. And this Puma chip actually works pretty well, and we're pretty hopeful. So the next part, what I want to show you really quickly, and so as you can tell, this talk is a little bit more technology intensive, but I think that's kind of nice, just so that we can show you some of the things we're trying to develop, some of the technology that enable one to do things, and then to get some input from there. So we have isolated those rare cells. The next question is, what do you do with the cells that you have isolated? You obviously want to get as much information about those cells as you can, because after all, they are not so easy to isolate, but once you have them, you want to do a lot with it, right? If it's a fetal cell, in principle, that you have all the information, at least the genomic information about a fetus. So if you can do DNA sequencing out of that one cell, that would be great. So some of those are the things that we're thinking about, and some of them can be done right away, and some of them are more sophisticated. And I will show you 2 examples where one, I think, is pretty easy to adapt, more mature, and one, I think, is a little bit further down the road, but it's still exciting. So I'm going to tell you 2 short stories there just to give you a flavor, and then there are a lot of other things we work sort of in between. So actually, before I talk about the second part, I just want to thank the people that have done all the work over the years. Perry and Mengsha has done a lot of the work on the platform that I told you about, as well as Jason, Thomas, and many others over the years and joining the group. We are really very happy and fortunate to be able to work with very good collaborators and learn from them. So Dan, we've been working with, and we share a grant for 3 years, and as well as Qianhua. So many of the data that I show you is a result of this collaboration, and also the people in their lab that has done many of those work, and the people that have funded us. So the next part I want to show you really quickly is that one of the things that's a little bit more mature is to do gene expression profiling, PCR, because you can do single cell PCR quite easily. And in fact, there's commercial kits and so on and so forth. And we do use them. We can isolate those cells, put them into a small Eppendorf tube. You can do qPCR, things of that sort. But we're also trying to develop a microfluidic approach that we hope is more efficient, where instead of picking it up and then putting in a tube and then put in another tube, we want to have a device that can do higher throughput single cell gene expression analysis and so on and so forth. And so, so this is a chip that we have been working on, and I just want to show you what it can do, just a very quick overview of it. But anyway, the main question, I suppose, is that when you try to do single cell PCR, what is the most difficult step? Is it detecting the product is, actually it's not none of those because you can amplify things. The most difficult part, especially if you have many cells, is to put one cell into one small volume so that you can amplify in that small volume, the products are concentrated, and then you can detect it, right? And we do a lot of microfluidics, and it turns out that we found out of discover a phenomenon that we can actually do this spontaneously very easily. So we have a chip that looks like this, lined with a bunch of nanochambers, about a nanoliter or so. And it turns out that the shape of the chamber is such that if you prime this chip with oil, like mineral oil or whatever you have, and that's very easy to do. You just pipette it into the reservoirs, you put it in a desiccator or something, it goes in and you prime it. You can put it aside and you can store it. Then if you put if you pipette in some aqueous solution, could be cell solutions, could be other things, and then apply a pressure to drive it in, it turns out that that aqueous solution would like to go into those nanochambers and form tiny little drops in those chambers. So this is what it looks like. So if it's primed with oil and you have aqueous solution that go through, it actually just sort of digitizes into an array of those tiny little drops. So we found that, hey, could be very useful because if you have individual cells that can go into those chambers, then you can do actually single cell analysis, gene expression analysis at least very easily. So this is the idea, and we are very interested to sort of further develop this because it's really easy to use. If you want to do very small chambers using microfluidics with valves and pumps, that can be quite complicated, but this is really easy. All you have to do is pipette in, and then we have a double-sided tape with a piece of plastic that we hook it up to a pressure source, and then it drives in, and then you digitize this. And the nice thing is that you can digitize every single bit of the volume without any loss. So if you worry about losing cells, or if you have very limited samples, you can actually digitize everything. So this is what it looks like. As long as the capacities of those nanochambers are greater, the total volume of the nanochambers add together is greater than your sample volume, everything gets digitized. And the other thing that is very nice is that if you, let's say, do PCR and saw a certain set of genes, and you say, hey, okay, because you have those genes, we want to do a closer look, you can in fact remove those volumes, and then you can think about maybe do a whole genome amplification or transcriptome analysis and stuff like that. So you can actually also remove things. And so once I won't go into the fluidics of it, but once you have done it, you can actually design all kinds of geometries to create higher densities and so on and so forth. And so this is what we have done, and this is one of the things that we're doing towards single-cell gene expression analysis. And in fact, we got them to work. And so one of the directions that we've been looking at is actually low copy genes, because many of the things that are really interesting are present at low copies. They're not housekeeping genes. So in that case, if your copy number is very low, it's often very difficult to use real-time PCR because you have uncertainties if your threshold cycle is not quite up to spec. So one of the things that people are very interested in doing digital PCR, and in fact you can do that in this chip. We can do digital PCR, and in this particular case it's called digital lamp. So it's not PCR, it's an extra isothermal amplification, and you can do digital lamp and things of that sort. So not only potentially you can do single cell analysis, you can in fact think about dividing the content of the cell to do digital analysis of gene expression and things of that sort. So this is one direction that we're working on, and because it is based on very well established PCR, but in a different format, this one is actually, I think, a little bit more mature than the other thing I'm going to tell you about. And so the other thing that I want to tell you about is further down the road, although we have worked on it for quite some time. And that is, if you think about a cell is actually very small, 10 micron, 20 micron, whatever you have. And so it's quite small, but at the same time, it's really complex. It's got organelles, just like an organism that has organs, cells, has all kinds of organelles. There's spatial, temporal compartmentalizations of molecules and signalings and so on. So one thing that we wanted to do is that, well, if we only have a few cells, and every single cell is different, we really want to look at this one cell and we want to study how it is, then the analogy is kind of like before. We want to basically do surgeries in the cells, isolate things that we want to further analyze particular regions of the cell without damaging the cell too much. We want to be able to image the cell to see what proteins are expressed and so on without destroying the cells. And so this is something that we are trying to do. And so there are actually different pieces to this. The basic idea is that we're going to take a cell, we're going to study it with high resolution microscopy without damaging the cells. Those are live cells. And then we would develop surgery techniques, but not on organisms, but on an individual cell, surgical techniques, so that we can isolate small pieces of the cell without affecting the cell too much. And so this is done using lasers. So we have something called single-cell nanosurgery that we have developed over the years. Here's just an example, and these are different examples I'll show you. Here we can pull out an organelle from the cell here, and this is probably a few hundred microns, and then you can move it. And you can actually use laser to move things because laser has photons, and photons, when they get bended or when they get bounced back, has recoil momentums and stuff like that. So you can actually use that to move things and to do surgeries on individual cells. Here's done a little bit differently, where we use one laser to poke a hole, and then we use another laser to drag out and pull out an organelle. And if you let it go, you can see that it disappears because it's very small. It's about probably a couple hundred nanometers. And we are combining this with, not in this video, but we're combining this with fluorescence so that you can actually use molecular recognition to say, some labeling tags or something to actually see what you are taking out. And at this moment in time, we didn't know what to do with it, so we can actually put it back. And we characterize the cells, and the cells are actually fine. Most of the time, the cells are fine. So we study different cell lines and study them. And in retrospect, I guess it's not too too surprising, because you're taking a small thing out of the cell. And after all, if you look at what people do to a cell when they clone things, they take a pipette, they suck out the nucleus, they spit it out and stick it in another nucleus, and then the cell grows into a sheep, right? So they're pretty okay with losing a few organelles. The nice thing with those optical techniques is that you can actually parallelize them. We are not limited to one thing at a time. If you're sort of doing a surgery, you have a pair of hands, but optics, you don't. You can have tens and hundreds of pairs of times. So we do things like using holograms to create many, active regions, so we can do many things at a time to actively manipulate. So we're trying to do surgery not with one organelle, maybe with 50 at a time, things of that sort, in real time. And the other thing is that once we have isolated those organelles, we also want to study them, right? If you study things that are that small, what do? You need to do chemistry, you need to do biochemistry, you need to do assays. So we have also been developing a way to do chemistry at very small length scales and volume scales. So instead of doing things in the lab with tubes, there are milliliters, microliters. We're trying to shrink it maybe 1 trillion times in volume to volumes that are femtoliters, like 10 to the minus 15 liters. And so the way we do them is using droplets that we can make on demand, and then manipulate and do additional things with it later on. So this is an example. We have developed different techniques. This is one technique that we developed, that we can press a button and then make a droplet on demand. And these are droplets. This is 2 micron. So 2 micron is about the length of of an E. coli, for example. And if you look at a red blood cell, that will be 6 microns. So they're relatively small length scales that works well with organelles and really small pieces of stuff you get from the cell. And combining with optical techniques, we can trap small things, and then we can use fluidics to spit them out, and then we can put them into those very small containers and start thinking about doing chemistry, biology, chemistry, and so on. And they won't come out because outside is oil, inside is water, unless you fuse it with another interface that releases them back into aqueous solution. We developed those things called vortex traps for fusing those droplets, because you have to pick them up somehow. And so we actually use optics to pick them up and then put them together, so you can do chemistry and initiate chemistry in very small length scales. And here are examples of organelles, mitochondrion, one mitochondria, a single cell. You can do all kinds of single cell assays and organelle assays you can think about. And then if we release them into aqueous solution, then we can also try to read out what the contents of those organelles are. And to do that, I won't go into this too much detail. We use single molecule techniques so that we can separate molecule by molecule, and then looking at what their charge to mass ratio is, try to identify what they are, and so on and so forth. So this is something that we're still working on quite a bit. As you can see, this is going to take some time because it's not quite, it's not quite a walk in the park. But ideally, what we like down the road is that we're going to have a platform so that we have only a few cells that we isolate from the patients. And if we really want to zoom in and study in really great detail what the cell is doing, then we can image them, we can study them, we can see how they behave, how they migrate, and then we can, we can use optical methods to interrogate them, to do nano-surgery, for example, and then do chemistry in those drops, and then read them out using single molecule techniques and things of that sort. And I heard Pete's phone bleep blip a couple minutes ago. That I think is a cue that I should finish up. And for this part, we've worked on it for quite a lot of years now, and so there's been people that have gone through the lab over the many years that have contributed, and I would like to thank them, and also the sponsors that have believed in us and supported us over the years. With that, I think my time is up. Whether instead of removing things, whether you can use that to introduce things. And yeah, we try some of those, and in fact, in one of the videos, we could, in fact, put some of the things that we're taking out back in. So I think that is possible, especially if you just want to get it into the cytoplasm. But getting into a nucleus, we haven't done it, and the nucleus is much, we just haven't tried, but it's got a double layer and it's very rigid structure, so I don't know how that will work. So we haven't done that yet.
So that was a beautiful talk, very impressive. My question relates to the first part of your talk. Once you do the size selection, then you could do flow cytometry, is that right?
Yeah, we call it Eedar, so it's not really flow cytometry because we're looking aliquot at a time. So we actually would do that first and then do the filtration down the road.
So you could stain the cells for whatever antibodies you want with different fluorochromes, and then you could do size selection and then select for expression of different.
Yeah, so basically we go through this aliquot ranking process, isolate the aliquot that we wanted, and then we further purify with the filtration. Yeah, so we would, yeah, we can use any markers that we want. As long as it's a good marker, then we can use that.
Yes, again, a very interesting talk. Thank you. Have you been able to look at cell-cell interactions on a single cell basis with rare cell populations interacting with each other in some way?
Yeah, that's really interesting. We haven't gone around to do that, but that would be, I think that would be really nice to do.
Yeah. Sure, like if you could isolate one cell in one of those little pockets and then just look at the interactions.
Yeah, I think that would be really, yeah, I think that would be really, I think we're all set up technically to do this. It won't be too difficult to do, but we haven't done it yet. And also, I should probably talk to you more and see what kind of questions that one might sort of zoom in on if one can have the capability of doing such a thing.